there is one place in your body where your brain is visible. Not on a scan, not in a laboratory, in your bathroom mirror, in a well-lit room, with nothing more than your own eyes and the knowledge of what to look for. The retina, the thin sheet of neural tissue lining the back of the eye, is not merely an optical sensor. It is a direct anatomical extension of the central nervous system. The same blood vessels, the same neurons, the same glial cells, the same amyloid proteins that accumulate in the aging and degenerating brain are present in the retina, accessible to examination, and changing in measurable and specific ways years before the brain produces any symptom that a person would notice or report, before the memory lapses begin, before the processing slows, before any cognitive test shows a decline. The retina is already showing the evidence. Research published in the last decade, including landmark studies from the UK Biobank involving over 50,000 participants from the Alzheimer's Disease Neuroimaging Initiative and from multiple independent retinal imaging cohorts, has established that specific observable changes in the retinal vasculature, the optic nerve, and the retinal nerve fiber layer predict the development of Alzheimer's disease, vascular dementia, and other neurodegenerative conditions with a lead time of 5 to 20 years before clinical diagnosis. 5 to 20 years, the window in which intervention is most effective, the window that most people spend unaware that the window exists. I am Dr. Edward. What you are about to hear is educational information based on published scientific research. It is not a substitute for personalized medical care. If you are experiencing changes in vision, memory, or cognitive function, please see both your ophthalmologist and your physician. The retinal signs I am going to describe are clinical findings that require professional evaluation to identify and interpret. I am giving you the framework to know what questions to ask, not to replace a clinical examination. Subscribe to this channel right now before we go further. The research I am about to describe represents one of the most significant developments in preventive neurology of the last decade, and virtually none of it has reached the general public in a form they can act on. Subscribe now. It is free. And stay with me for the full picture. Let me start by explaining why the eye has this unique relationship with the brain, because most people think of the eye as a camera attached to the brain, rather than as a piece of the brain itself. And that distinction changes everything about how we interpret what happens to the retina as we age. During embryonic development, the retina forms as an outgrowth of the developing brain, an evagination of the diencephalon, the same region that develops into the thalamus and hypothalamus. The optic cup that forms the retina carries with it the cellular composition of central nervous system tissue, neurons, Muller glial cells, astrocytes, and microglial cells. The retinal ganglion cells, the neurons whose axons bundle together to form the optic nerve and carry visual signals to the visual cortex, are the same class of projection neurons that connect brain regions to each other throughout the central nervous system. The blood vessels that supply the retina share the same developmental origin and the same structural characteristics as the small blood vessels of the brain. The blood-retinal barrier and the blood-brain barrier are built on the same cellular principles and respond to the same pathological stressors in the same ways. This developmental and structural continuity means that what happens in the brain happens in the retina, not as a secondary consequence or a distant reflection, simultaneously through shared pathology, because the retina is part of the same neural system. When amyloid beta, the protein that aggregates into the plaques associated with Alzheimer's disease, accumulates in the brain, it accumulates in the retina at the same time, depositing in the retinal ganglion cell layer and in the walls of retinal blood vessels. When the small vessel disease that underlies vascular dementia begins damaging the brain's microvasculature, the same disease process is simultaneously damaging the retinal microvasculature, where it can be directly visualized. When the neuroinflammation that drives tau pathology in Alzheimer's activates microglial cells in the brain, the same microglial activation is occurring in the retina. The eye is not a window into the brain in a poetic sense. It is a window into the brain in a literal anatomical sense. And until recently, we lacked the imaging technology to look through that window with sufficient precision to extract the information that was always there. That has changed. The first retinal sign of brain aging that I want you to understand is thinning of the retinal nerve fiber layer, the RNFL. The retinal nerve fiber layer is the innermost layer of the retina, composed of the axons of retinal ganglion cells as they converge toward the optic disc to form the optic nerve. In a healthy retina, this layer has a measurable thickness, typically between 90 and 110 micrometers at the peripapillary region, the ring of retina immediately surrounding the optic disc. As the ganglion cells die, whether from Alzheimer's pathology, from vascular disease, from chronic elevated intraocular pressure, or from the normal attrition of aging, the RNFL thins. That thinning is measurable with a technology called optical coherence tomography, or OCT, which uses near-infrared light to produce cross-sectional images of the retinal layers with micrometer resolution. OCT is available in virtually every ophthalmology practice and many optometry offices, often performed as a routine part of a comprehensive eye exam. A meta-analysis published in the journal Ophthalmology, analyzing 30 independent studies, found that RNFL thickness in the peripapillary region was significantly reduced in patients with Alzheimer's disease compared to healthy age-matched controls, with the greatest thinning observed in the superior and inferior quadrants. More importantly, RNFL thinning was measurable in people with mild cognitive impairment, the stage between normal aging and dementia, and in people with subjective cognitive complaints before any objective cognitive decline was measurable on standard neuropsychological testing. The RNFL was showing the pathology before the cognitive tests could find it. The practical implication is this: if you are over 60 and you have not had an OCT of your retina in the last 2 years, ask for one at your next eye appointment. Ask specifically for a peripapillary RNFL analysis. This is a standard scan. It takes 3 minutes, it involves no discomfort, and the thickness maps it produces contain information about your brain's neuronal health that your annual physical blood panel cannot give you. Subscribe to this channel right now if you want to keep receiving clinical research that translates directly into the questions you ask at your next appointment. This is exactly what I do every week. Subscribe. It is free. And let us continue. The second retinal sign is changes in the retinal vasculature, the network of arteries and veins that supply blood to the retinal tissue. Retinal vascular changes are perhaps the most extensively studied retinal marker of systemic and neurological aging, and for good reason. The retinal vessels are the only blood vessels in the human body that can be directly observed and photographed without surgery or invasive imaging. A fundus photograph, a standard image of the retina taken through the dilated pupil, captures the entire retinal vascular network in a single image that can be analyzed for vessel caliber, tortuosity, arteriovenous ratio, and vascular wall changes. The retinal vascular findings associated with brain aging fall into several categories. The first is arteriolar narrowing, a reduction in the diameter of the small retinal arteries that reflects the same hypertensive and arteriosclerotic changes occurring simultaneously in the brain's small vessels. The arteriolar to venular ratio, the relative diameter of arteries compared to veins, is a well-validated marker of systemic small vessel disease. A narrowed arteriolar to venular ratio in the retina is independently predictive of white matter hyperintensities in the brain on MRI, of lacunar infarcts, small silent strokes, and of cognitive decline. A study published in JAMA Neurology followed over 2,000 adults for 15 years and found that those in the lowest quartile of retinal arteriolar to venular ratio at baseline had a 47% higher risk of developing dementia over the follow-up period compared to those in the highest quartile, after adjusting for all other vascular risk factors. The second retinal vascular finding is arteriovenous nicking, the visible compression of a retinal vein at the point where a retinal artery crosses over it. In the healthy retina, arteries and veins share a common adventitial sheath at crossing points, and neither vessel compresses the other. When the arterial wall thickens from arteriosclerosis, the arterial stiffening that occurs with chronic hypertension and aging, the rigid artery compresses the more compliant vein beneath it, producing the visible kinking or deflection of the vein that ophthalmologists grade as AV nicking. AV nicking is a marker of arterial wall disease and is associated with increased stroke risk, white matter changes, and vascular dementia. It is visible on a standard dilated fundus exam by any ophthalmologist. The third retinal vascular finding is the presence of retinal microaneurysms, or dot hemorrhages, tiny outpouchings of retinal capillary walls, or small hemorrhages from capillary breakage. These findings, most commonly associated with diabetic retinopathy, also occur in non-diabetics with chronic hypertension and metabolic syndrome, and they represent the same capillary failure occurring simultaneously in the brain, the same process that produces the cerebral microbleeds visible on brain MRI in people with chronic small vessel disease. Their presence in a non-diabetic senior is a marker of systemic microvascular compromise that warrants both aggressive cardiovascular risk factor management and, increasingly, neurological evaluation. Now subscribe right now if you want to make sure the follow-up video on retinal imaging, where I walk through what an actual OCT report looks like and what to ask your ophthalmologist when you receive one, reaches you the week I release it. Subscribe, it is free, and it takes 2 seconds. The third retinal sign is changes in the optic nerve head, specifically changes in the morphology of the optic disc that reflect the health of the ganglion cells projecting through it. The optic disc is the point where the retinal nerve fiber layer converges and exits the eye as the optic nerve. In a healthy optic disc, the central cup, the physiological depression in the center of the disc, occupies a proportion of the total disc area, expressed as the cup-to-disc ratio. A cup-to-disc ratio above approximately 0.6 is associated with glaucoma, the most common optic nerve disease in seniors, in which elevated intraocular pressure damages ganglion cells and causes cup enlargement. The significance of glaucoma in this context is not just as an eye disease, but as a neurodegenerative condition sharing pathological mechanisms with Alzheimer's disease. Research has found that patients with primary open-angle glaucoma have significantly elevated rates of Alzheimer's disease diagnosis compared to age-matched non-glaucoma controls, and that the mechanisms driving ganglion cell loss in glaucoma, neuroinflammation, amyloid accumulation, mitochondrial dysfunction in neurons, disrupted axonal transport, are the same mechanisms operating in Alzheimer's neurodegeneration. Glaucoma and Alzheimer's may represent the same neurodegenerative process expressed in different primary locations depending on where the pathological vulnerability is greatest in that individual. This means that a glaucoma diagnosis is not merely a vision management issue. It is a neurological risk marker that warrants discussion with a neurologist about cognitive monitoring and early intervention strategies, a conversation that almost never happens because ophthalmologists and neurologists rarely share patients or communicate about shared pathology. The fourth retinal sign is drusen, the yellowish deposits that accumulate beneath the retinal pigment epithelium in age-related macular degeneration. I want to address drusen specifically because AMD affects approximately 11 million Americans and is the leading cause of vision loss in adults over 65, but its neurological implications are almost never discussed outside of ophthalmology. Drusen contains several components that are directly relevant to the brain aging discussion. They contain amyloid beta, the same protein that aggregates in Alzheimer's plaques. They contain complement system proteins, the same inflammatory proteins elevated in Alzheimer's disease and associated with neurodegeneration. And the drusen of AMD formed through a process called subretinal inflammation that shares mechanistic features with the neuroinflammatory processes driving Alzheimer's, Parkinson's, and frontotemporal dementia. A growing body of research, including work published in PNAS, has proposed that AMD and Alzheimer's disease share a common underlying pathobiology of amyloid accumulation and complement-mediated neuroinflammation, with the disease expressing primarily in the retina in AMD and primarily in the cortex in Alzheimer's, depending on which tissue is most genetically vulnerable. The practical implication is that a senior with AMD who does not have dementia should understand that their AMD diagnosis represents an active amyloid and neuroinflammatory process that is not confined to the retina. The systemic interventions that reduce amyloid burden and neuroinflammation, which I will cover in a future video, are relevant to their overall neurological risk in a way that most ophthalmology practices do not have the context or time to explain. I also want to address what is perhaps the most exciting development in this entire field, which is the application of artificial intelligence to retinal imaging for brain age prediction. Several groups, including a team at DeepMind working with the UK Biobank dataset, have trained neural networks on retinal fundus photographs to predict biological brain age, the age of the brain as estimated from its structural health rather than the person's calendar age, with remarkable accuracy. In the DeepMind research, the AI model predicted biological brain age from retinal photographs alone with an accuracy that correlated strongly with brain age predicted from actual MRI scans. The retinal photograph contained as much neurological age information as a brain imaging study. These AI retinal age gap tools, the difference between your predicted biological brain age and your actual age derived from a retinal photograph, are now being validated in multiple clinical cohorts as predictive markers of dementia risk, cardiovascular mortality, and all-cause mortality. They are not yet available in standard clinical practice, but several companies are moving toward clinical deployment, and within the next several years, a retinal photograph at your annual eye exam may automatically generate a biological brain age estimate alongside your prescription update. The retina is becoming the most accessible and information-dense diagnostic surface in preventive medicine. Now, I want to give you the practical action framework because the research I have described translates into a specific and actionable set of things to do at your next eye appointment that most seniors are not currently doing. The first is to request a comprehensive dilated fundus examination, not just the quick undulated photo that some practices use for routine screening, but a full dilated exam in which the ophthalmologist or optometrist directly views the retinal vasculature, optic disc, and macular region through the dilated pupil. This takes approximately 20 minutes including dilation waiting time and provides the direct visualization of AV nicking, microaneurysms, drusen, and optic disc morphology that I described. The second is to request an OCT, optical coherence tomography, with specific peripapillary RNFL analysis. Ask for the report to include the RNFL thickness map and the normative database comparison, which shows your RNFL thickness color-coded against the age-matched normal range. Values in the yellow or red range in the superior or inferior quadrants deserve specific discussion. If your practice does not have OCT equipment, ask for a referral to a practice that does. OCT is the standard of care in ophthalmology, and most practices of any size have it. The third is to request that your eye exam report be shared with your primary care physician. Most ophthalmologists and primary care physicians practice in complete isolation from each other, despite examining aspects of the same vascular and neurological system. A retinal finding of AV nicking, RNFL thinning, or significant drusen burden should be in the hands of your primary care physician, who is managing your blood pressure, your cholesterol, your metabolic health, the systemic risk factors that are driving the retinal pathology. The fourth is to ensure your blood pressure is optimally controlled. Of all the systemic factors that drive retinal vascular aging and the neurological aging it predicts, blood pressure is the most modifiable and the one with the strongest evidence for benefit from treatment. A systolic blood pressure consistently above 130 mmHg is driving arteriolar narrowing, AV nicking, and white matter damage in the brain simultaneously. Every point of systolic reduction from optimal management translates into less retinal vascular disease, less white matter damage, and measurably lower dementia risk. This is the most direct application of everything I have described today. Here is the summary of everything we covered. The retina is an anatomical extension of the central nervous system, sharing developmental origin, cellular composition, and vascular structure with the brain. Retinal changes associated with brain aging appear 5 to 20 years before cognitive symptoms, representing the most accessible early warning window available in preventive neurology. 4 specific retinal signs carry neurological significance. RNFL thinning measured by OCT, reflecting ganglion cell loss from Alzheimer's or vascular pathology, retinal vascular changes including arteriolar narrowing, AV nicking, and microaneurysms, reflecting the same small vessel disease driving vascular dementia. Optic disc changes from glaucoma, which shares neurodegenerative mechanisms with Alzheimer's disease, and drusen from AMD, which contain amyloid beta and complement proteins linking AMD to the same neuroinflammatory biology as Alzheimer's. The clinical actions to take are a comprehensive dilated fundus exam, an OCT with peripapillary RNFL analysis, communication of findings to your primary care physician, and optimal blood pressure management. AI retinal age prediction is emerging as the most accessible brain age biomarker in clinical medicine. Please share this video. There are seniors in your life who go to annual eye appointments that check their prescription and look for glaucoma and tell them their eyes are fine, and who have never been told that the tissue behind their eye is showing information about their brain that a brain MRI might not yet reveal. Share this with them and give them the questions that could change what happens at their next appointment. Subscribe to this channel if you have not already. My next video covers the specific dietary and lifestyle interventions with the strongest evidence for protecting the retinal vasculature, reducing amyloid accumulation in retinal tissue, and supporting ganglion cell survival. The cellular level interventions that address the mechanisms I described today. Subscribe so it comes directly to you. I am Dr. Edward. Your eyes have been showing you your brain for years. Now you know how to read what they are saying. I will see you in the next one.